not least, in our session today, another student in my lab, Sydney Stanton. Sydney is, as you will see, has created a really neat project. So I, of course, am an ecologist. I run around in swamps. That's my home. Sydney is going to be heading off to veterinary school, and so developed this really neat project that sort of combined, working with a swamp creature with her interest in more biomedical focused research. So it's been a real pleasure to work with her, and I will turn it over to Sydney to tell you about assessment of the antibacterial properties and skin microbiota of unisexual investoma salamanders upon secretion of defensive toxins.
All right, welcome everybody. So today I'm going to give you a talk about something you might not hear again, which is the experience of somebody that willingly played with mucus for over a year. So, yep, that's me in the field with one of our guys this year. So the main reason I did this project is to combat the issue of antibiotic resistance as they were discovered in 1928 with penicillin, clinically introduced in the 1940s, right in time for World War II. And then as we got into the 1950s and 60s, as these drugs are being used more and more, we saw the emergence of more antibiotic resistance, and it became more of a question of public health at that point. And it's gotten to the point where in 2015, the World Health Organization declared it a global emergency, and now we're facing projections where common injuries and infections could turn fatal if we no longer have effective treatments. And I was specifically looking at the escape pathogens here, which are some of the most common in hospital settings and are pretty deadly nowadays. And mostly because bacteria are so adaptive, they have antibiotic resistant genes that they can swap with each other and pick up from the environment. And the inappropriate use of antibiotics for things such as viruses has really not helped either. And this has led to about $20 billion in public health costs and 23,000 deaths per year from antibiotic resistant infections in the United States. And this is propagated heavily by environmental waste from pharmaceuticals, hospitals, and just anywhere where these antibiotic-resistant genes or escape pathogens can be found. So a solution for this, someplace that may have potential treatments for these infections, are in amphibians. They have a very complex immune system, and compared to most other vertebrates, they have permeable mucosal skin, which is pretty unique. And they have a variety of structural and molecular devices to protect them. As you can see here, they have different glands that secrete immune cells, neurotoxins such as tetrodotoxin for predator defense, and antimicrobial peptides. And there have been multiple studies that have shown that antimicrobial peptides, the microbiome, and tetrodotoxin can all interact and influence their prevalence in populations. So I won't go too much into kleptogenesis since we just heard a lot about that, but this is kind of an additional diagram showing how they can do ploidy elevation, genome replacement, organogenesis. Unisexuals are pretty unique and haven't had a whole lot of research done on their mucus or antimicrobial peptides, microbiome, or really anything like that. This is one of the ones that I swapped right here, being really cute. So most of the salamanders that I worked with had either laterale or jeffersonianum genomes. I didn't get any tigers. So my research questions is ambystoma mucus effective at inhibiting the growth of escape pathogen relatives? And also, what are some of the dominant bacterial taxa present in their microbiome? So in order to do this, we set out traps at the Edwin S. George Reserve overnight, took a tissue sample, which made them pretty unhappy, and they secreted some mucus at us as a defense mechanism. So we swabbed all over the glove and on the salamander there along the back, and we stored those tubes in sterile water. Now to get to the second part, where I tested them against the different escape pathogens, I took some of the escape pathogens that we have in storage here, struck them out for isolation to get discrete colonies, and then from those isolated colonies I made bacterial lawns on which I plotted the mucus samples distilled in distilled water. Oops. Okay, so here's a diagram of how I did that. So this would be the same pathogen. Spots 1 through 22 correspond to salamanders. Spot 23 is for distilled water, and then these two, 24 and 25, those were spots including pond water control samples. And I put about 10 microliters of each sample on one of those spots. Now, when I was looking at the results here, this is what I had hoped to see, which is a zone of inhibition. For example, like these are antibiotic discs, and they would inhibit the growth of bacteria around them because they're an antibiotic. And as you can see in the picture on the left, I really did not see any of that. There was no inhibition of growth by the salamander mucus against these escape pathogens. But there was a whole bunch of interesting stuff that grew from them, as you can see. So that was kind of the next part of my question was, what are these things? So in order to do that, I scraped off all the new bacteria that I found, put those in glycerol. They went through freezing for quite a while. But when I re-isolated them, I put them on R2A media, which supports slow growth of freshwater pathogens, different microbes. And after that, I extracted the DNA and subjected it to PCR, amplifying the 16S rRNA gene, which is basically DNA, the DNA name tag for bacteria. It kind of tells us what their identity is. So I made a library of their genes, sent those out for sequencing, and then compared it to our known sequence database for different bacteria called BLAST, basic local alignment search tool, where I tried to align and match these bacteria to what we know about others. So these are the identities that I came up with that matched our database so far. The highlighted ones are those that were unique to the salamanders and not found in the pond water. So basically everything was Pseudomonas, which was interesting to see. As you can see, there is Cyrtobacterium plantarum, which was actually isolated from soybeans in the 80s. That's when that was first discovered. So that was found in the bacteria in the pond, which isn't surprising. There's a bunch of lush plant life there. But Pseudomonas is interesting, and it makes sense that it survived, the freezing process, competition with escape pathogens, because they can do a lot with their metabolism. They can metabolize so many different compounds and survive a lot of stuff. They're a strong species, or group of species, that is. So kind of going into the conclusion here, Ambystoma mucus was ineffective, and so at inhibiting the growth of escape pathogens, so is likely not a good treatment for those that we know of. However, other studies have shown some antimicrobial defense, although it is limited. They've been able to extract and synthesize some peptides that have had anti-escape properties. And additionally, Pseudomonas has been described previously as part of amphibian microbiomes, and they can be anti-chytrid fungus, which is a disease wiping out so many amphibian populations worldwide, and also they can produce tetrodotoxin, some species, which basically in predators, it will bind to their sodium muscular and neural channels and cause paralysis. And we really don't have any evidence so far that the unisexuals can produce this. So while this isn't hard evidence, since we found Pseudomonas there and they may produce tetrodotoxin, it could be a potential avenue of research now that they have that in their system as a potential. So for future research, I would recommend, based on my findings, increased antimicrobial peptide extraction and synthesis to see what kinds of defenses these amphibians have against both amphibian and human pathogens, and also high throughput sequencing and rinsing when doing this type of procedure would help in retrospect to get environmental debris off and obtain a higher diversity of bacteria in the samples. So just some acknowledgements and shout outs. Obviously the herpetology lab, Dr. Greenwald, everybody for helping me with this project. Bio 111 at lab and Dr. Angel for supplying the microbiology side of this. Dr. Leitman and Dr. Judd for helping me with presentation, thesis writing. Dr. Goodrich and Dr. Casper also for helping with the microbiology side of stuff sometimes. And then everybody, of course, that came out to see my project. So here are some cute salamander pictures for the rest of your day. Thank you.
Awesome. Well, we have time for a few questions for Sydney, and this is the end of our session. So
Yes.
How was your sequencing done?
So essentially, I extracted and amplified the characteristic gene, and once I had those amplified, they were cleaned up and they were sent out for sequencing. So they sent those sequences back to me, which is where I compared them to the library of bacterial sequences we have so far.
Was it the Sanger sequencing?
Yeah.
Okay.
Oh.
We're so polite, we'll take all day. That's good.
I forget my question. Have you tried getting DNA directly from the swabs? This was indirect, right? Like you.
Yeah, they had to go through the whole culturing process. That is very expensive, so I wasn't able to do that. So yeah, the bacteria that I isolated, they did have to undergo culturing and freezing, so it is a bit more selective with the bacteria that I obtained, for sure.
Lovely though, that's what waves do.
Yes.
So, amphibians, of course, have their two lives. The microbiome and the sort of infection challenges.
Yeah.
What would you think would be different about in the water and not so much in the water?
So I guess like, you mean in their environments versus the lab I did the experiment in?
No, but their life where they're clearly all in the water and their life when they're not so much in the water.
Oh, yes, yeah, so there are, like if I go back here, there are going to encounter different bacteria based on the pond or if they're in land. These samples were collected when they were in the ponds for their breeding season about a year ago. So a lot of their microbiome might have overlapped with the pond water since we didn't rinse them before the swabbing procedure. So since we got a lot of pseudomonas from the ponds as well, perhaps that was influencing their microbiome. I don't really have any evidence so far as to if this changes when they're living more terrestrial lives. But that would be a good avenue for future research.
All right. Well, that is the end of our session. Let's thank all of our speakers again.